Hello, students. Am I audible? Please confirm in chat box if I'm audible. Okay, great. Yes. Good evening to all of you. How are you all doing? Excited about the session? Super excited? All right. Then type a big BIO. Bring it on. And let's start with our rapid revision rounds. Yes, Biotecnika always comes with you at the last moment with how much ever help we can. And yet we are discussing with you the most important types of questions from Unit 1. Unit 1, yes, it's biochemistry, isn't it? Now let's see our first question. Are you ready? Yes, you have to answer me in chat box. Okay? And after you give me your answers, I will tell you the correct answer, and obviously the explanation for it also. I believe you have a decent preparation by now for biochemistry. And here comes on your screen the first question. Hill's equation and its plot. Describe the following enzyme kinetic behaviors. A. Saturation kinetics. B. Cooperative kinetics. C. Something versus something. X-axis versus y-axis. And something versus something. What's your question? Which one of the following combination represents correct descriptions? Your options are A and C, B and C, B and D, and A and D. Go ahead. So, Diksha, if it is blurred for you, you can just try to change the settings. There you can choose high quality, high definition. The quality over there you can choose in advanced 1080p. Yes, that's amazing. MT Nurjahan, Nida Parveen, Niharika Pandita. So, Vishal, you have to select any one of these four options. Pratibha, yes, very good, everyone. Your answers are right. Come on, others also can do it. So Hill's equation, it is, and the plot, it is to determine the ligand binding, the protein and the ligand binding. Yes, very good. Rimjim, Nivedita, Amisha, Nancy, Mukul, Nikhil, Ankita, Shashmita, Sadav, Sampurna, Nanashri, everyone. Correct. Very good. So here is your answer. A plot of log y divided by 1 minus y. This is what you take in your x-axis versus log L. This goes in your y-axis. This is what is your Hill plot, where n is the Hill coefficient. The equation is in the form of y is equal to mx plus c, mx plus b. So what is mx plus b? Yes, mx plus b is, this is a straight line equation, isn't it? Where slope n and y, intercept of log minus d. The intercept is of minus log d. Now when n is equal to 1, when slope is 1, as is with the case of myoglobin, which has only one ligand binding site, when oxygen binds to each site with the same affinity, irrespective of the number of oxygens bound to other sites, then the Hill plot is linear with a slope of 1. Log L values near the region of the curve that crosses the x-axis approximate a straight line with a slope of 2.8, and that will imply maximum cooperativity in the middle of the binding curve. So what is cooperativity? Cooperativity, let's say hemoglobin. Hemoglobin will be having 4 subunits. Now when a ligand binds on one subunit, the other subunit, first of all, this subunit will undergo some changes. Because of that, there will be a change even in the other subunit such that the ligand binding will become easier. Since these subunits are connected with each other, it's a quaternary structure. That is why even this will be changing the conformation, and so will be this. So isn't the ligand binding becoming helpful as more and more subunits are there? So this is called as positive cooperativity, where the binding of ligand is helping the binding of ligand on another subunit. So the graph, I'm going to show you the graph. It will show that the KD for the first oxygen binding is much higher. What is KD? Yes, all those who are having a proper preparation would be knowing KD is the dissociation constant. It is when a protein and a ligand is binding, they have some association constant called as Ka. The opposite of Ka is Kd. Now, Kd is inversely proportional to the affinity. Affinity means the ability of the protein and ligand to bind together. So, if the Kd for the first oxygen is higher, it means that the binding of the first oxygen is less. The affinity of binding is less than the Kd for the last oxygen binding. So Hill's plot always shows for cooperative kinetics, how easy it becomes as and when the ligands keep on binding. So this is your Hill equation. Note it down, everyone who haven't been able to answer the first question. This is what I was talking about. Log y divided by 1 minus y. What is this? That's what I mentioned. It is the slope. And what is this? Log s, what you take in x-axis. See, basically we have a straight line graph equation. We have our y-axis. We have our x-axis. And when we take this, we will take this in log y 1 minus y. And what we will take in our x-axis? We will take log of substrate, right? And this would be your intercept. Intercept means there would be some line which will be crossing the y-axis, isn't it? So this is the y-intercept. This point is what is called as y-intercept, and which is given as in this equation, it is minus log of Kd value. Okay, so by this equation you can see this y is what? Y is fractional saturation. How much of the proteins are able to bind, how much of the ligands are occupying the protein binding site. Means out of the total binding sites available, how many are getting attached with the ligand. Fractional saturation means what? Bonded. The protein ligand binding sites are bonded. That is when it will get saturated, but not completely. Only fraction of it will get saturated. That is what is fractional saturation. So, in terms of our enzymes, what is most saturated will be your Vmax, the maximum velocity, isn't it? So, that's what 1 minus y log Vmax, that is what is going to tell you how much the enzyme and the substrate will bind. See, binding kinetics is not much different than the ligand or the protein or the enzyme and the substrate. Just the words are different. Okay. So if you check this one, this is what you will have. The Y over here will be your VI, how much is binding, the fractional saturation. And when it is completely saturated, that will be Vmax, isn't it? What is 1? 1 is when out of, let's say, out of there are 100 proteins, out of that all 100 proteins binding sites are occupied by ligand. So what does it become? The fractional saturation, it becomes 1, isn't it? So at 1, obviously all the enzyme sites, if they are occupied with substrate, then only you will get the maximum velocity. All the enzymes are working at their maximum velocity. That is what is your Vmax. Vmax minus Vi. And in the y-axis and the x-axis, you will be writing log s. So yes, according to that answer, as you can see, it's cooperative kinetics. Behavior, it explains cooperative kinetics. And C, where you take Vi divided by Vmax minus Vi versus log S. Okay, ready for your next question? Yes, slide is blurred. You people can change your resolution setting to 1080p under quality, please, if the slide is blurred. Okay, ready for your next question. Next question on your screen. So, in a mitochondrial respiration experiment, a researcher observed the following profile of oxygen consumption upon addition of certain compounds, but they were added at separate times. So these are the, they have not, this is not in order, okay? This is just a list of what has been added. Now, which of the following describes this profile which is given in this graph correctly? So these are your options on the screen. Yes, please solve this question. So this question is from June 2016. We are discussing the last 5 years questions. I've picked up from almost all the question papers. Since we have limitation of time, only a few questions we'll be able to discuss. but I'll try my best here to make you understand. No, Sunanda, this is only in YouTube. So I believe you all are able to hear me. So where are your answers? Okay, take your time, no problem. Yes, very good. Manjula, your answer is right. Correct. Yes, Abdul Ghani, correct answer. Good choice. Panisha 96, very good, correct answer. Averi Ujwal, you are right, very good. So I'm really glad that some of you are giving me correct answer. Yes, Tisha, Mahima, Shashmita, Pratibha, Vishal, very good. Yes. And I'm very glad that you people are not hurrying into the answering. You are gradually first checking the accuracy of the question and then you are answering. So this is the correct approach which you should follow in exam also. Okay? Yes. So the correct answer over here is option 4. So first, let me just show you something. See, first of all, we know in Krebs cycle, you know TCA cycle, right? So there is an enzyme isocitrate dehydrogenase, correct? Which is inhibited by NADH and ATP but activated by ADP. Like that citrate synthesis also there, which is inhibited by citrate by ATP. Correct? So here at time 1, if you add ADP, now, ADP is an indication that there is no energy source, right? There is no energy, there is less energy. So, if you have ADP, it will make an increase in the mitochondrial respiration. The ATP synthesis will increase through oxidative phosphorylation. And that is why here, the first one, see, it's oxygen consumption. Why is this even a thing in the y-axis? Whenever you see any graph-based questions, you're supposed to look at y-axis and x-axis, what they have mentioned. So with passing of time, you can see that the oxygen consumption is getting increased. Now, how is that related with ETC? Who can tell me? With aerobic respiration, with oxidative phosphorylation, how is this oxygen consumption related with the time, with electron transport chain? Yes, tell me. Yes, hello Antara. No problem, Rizvi. I'm explaining over here. You can see the explanation. I'm sure you will be able to understand. Okay, so I'm not getting any answers yet. Oxygen consumption. How is even that a thing? So many of you have answered the correct option, but you should be knowing why also, isn't it? I hope you have not memorized the answers or something. So this is what is the terminal electron acceptor. Yes, very good. Nida Parveen. Correct. Nandini, right. Absolutely correct, Antara. Right. Oxygen is the terminal electron acceptor. Yes, Janvi Tisha also right. So as a result, tell me something. Correct me if I'm wrong. Tell me if I'm right. Oxygen consumption, if it is high, if oxygen consumption is high, so that means electron transport chain is proceeding. True or false? Oxygen consumption is high. That means the electron transport chain is proceeding forward. True or false? Yes? Yes, it's true. Very good, everyone. Yes, it's the true thing, isn't it? So that is why you can see that there is no oxygen consumption here. No oxygen consumption here, isn't it? You can see it is a flat line. Now, after addition of something you have added in here, because of which suddenly the oxygen consumption increases. That too by leaps and bounds. So why did this thing happen? Because you have added something which has activated the respiration. which has activated the electron transport chain, which has started the electron transport chain. So you have this succinate and ADP and PI as activators, because succinate is an electron source and ADP and PI are activators of enzymes of the electron transport chain. Okay, so if are activators of the Krebs cycle enzymes. So if Krebs cycle enzymes are activated, even the electron transport chain will be activated. Now we will go with A, because in most of this is A, and that's why we will consider the majority. We will see if we don't get this correct, then we will pick succinate. So that's what you should do, right? 1A means out of this which are activators. These two are activators. So in most of them you have A. So better select A only as option. So 1A is fixed. Fine. Now after something you have added, after the oxygen consumption has increased, electron transport chain has proceeded forward, you see that again it has kind of fallen flat. Not completely flat, but it has come down a lot. So that means what? Something is either, it's not completely stopping the electron transport chain. So that means what? We know H plus ions are accommodated, because of which the ATP synthesis happens, isn't it? But something is stopping the ATP synthesis. So, out of your what can stop the ATP synthesis? Oligomycin. It is an ATPase inhibitor. Now, when you have a mitochondrial membrane, this is the outer mitochondrial membrane, this is the inner mitochondrial membrane, this is the matrix, let's say. So, from here, H plus ions are transported in the intermembrane space. Now, if these H plus ions are coming back to the matrix, then ATP is produced. But what if these are not able to come back? That means the enzyme ATP synthase is blocked. That is why they are not able to come back. That means this second you must have put C over there. Okay. And what is it? What are you adding in 3? Now see, even if the electron transport had slowed down, first of all, why did it slow down? Because there is a limit to how much H plus ions you can add, isn't it? There's a limit to which H plus ions can be transported. They will reach saturation on the other side. So that's why it slowed down. But something you added in 3, which again took it upwards, right? So that means the H plus ions gradient, it is not able to come back through the ATP synthase because it is inhibited. That means they are being taken through another source. So which is that another source? Dinitrophenol. What is it? It's an uncoupler. They break the connection between ETC and the ATP synthesis. Generally, these two processes are linked. They are coupled together. The electron transport chain will send H plus, H plus will come and produce ATP. But ATP synthesis is blocked, H plus will accommodate. So dinitrophenol or DNP will somehow take out those H plus from the other side. They will sequester those H plus, thus taking out the gradient. So again the electron transport chain can proceed. Okay, clear all those who couldn't solve? Light bulb moment. Did you understand this? Right? So dinitrophenol act as uncoupling agent when added to cells. They stop ATP synthesis, but electron transport still continues, so oxygen is still consumed. Now this is your these are your inhibitors and these are your substrates. You need to learn them. Okay, for example, rotenone, it inhibits complex 1 along with amytal. Then complex 2 is blocked by antimycin. Complex 3 is blocked by antimycin. Complex 4 is blocked by cyanide is there. Then carbon monoxide is there. Remember, all these are blockers of the complex 4. Even azide. Okay, these are all blockers of complex 4. You need to remember these for some other type of questions. And oligomycin, I already mentioned, it inhibits ATP synthase. Malonate, it inhibits complex 2. It is an inhibitor. But you have activators also: succinate, ascorbate, glutamate, malate. All these are activators. All right. Okay. Next question. The following are 4 statements on peptide or protein conformation. Glycine has the largest area of conformationally allowed space in the Ramachandran plot of phi and psi. Then a 20 residue peptide that is acetylated at N terminus and amidated at C terminus has phi values of minus 60 and psi values of minus 30 plus minus 5. for all the residues. It can be concluded that conformation of the peptide is helix, strand, helix. Third, the allowed values of phi psi are not valid for short peptides. Fourth, a peptide acetyl A1, A2, A3, A4, CONH2, basically it's a tetrapeptide, adopts a well-defined beta turn. The dihedral angles of amino acid second and amino acid third determine the type of beta turn. Okay? So choose the combination of correct statements. A and B, B and C, A and D, and C and D. Come on, answer it. So this is from December 2016, and henceforth Ramachandran plot keeps on coming. In questions, it keeps on coming. So read option B carefully. I'm getting few answers. Yes, Ujjal, your answer is right. Yasira, Kantimati, Janvi, Nancy, Kavya, Devanshi, Hiral, Amrita, Explore Life Sciences, Ankita, Sunanda. Yes, all of your answers are right. Nandini, right. Hunter Bapu, C is not an option. You have to select 1, 2, 3, 4. Yes. Deepa, right. Romy, correct. Yes. All of you whom I took out the names, those are right. So A and D is the right answer. Now let's see why. Okay. First of all, I hope you would be knowing about Ramachandran plot and about little bit about amino acids. Glycine is the simplest amino acids with no side chains. So no steric hindrance occurs while the rotation of bonds. Now what bonds I'm talking about? A little bit of the peptide bond. So see, this is a peptide plane. Okay, it cannot rotate. It does not have freedom of rotation, this CN bond. Similarly, this CN bond also does not have freedom of rotation. What can rotate is these single bonds. That is this phi value and this psi value. Now, because of their rotation, the entire polypeptide backbone, so many phi psi values will be there that they will rotate and they might take different kind of structures, the backbone structures. So, which are called as the conformation of the peptide. When you have a polypeptide chain, this polypeptide chain, it might fall into some alpha helix, some beta sheet, some beta turn, some loops. So, like that, you will have a folded polypeptide, which is the native protein structure. So how does it fold? Because of this phi and psi dihedral angles, right? So the in case of glycine, since it's a very small amino acid, the rotation is a lot. So that's why you will have a lot of options. The dihedral angles will be able to rotate in many angles. Hence, a lot of conformations can be taken by the peptide which contains glycine. So the first statement is a right statement. Glycine has the largest area of conformationally allowed space. Conformationally allowed means it will be having lot of conformation choices. Then second one, it says acetylated at end terminus. This is a common modification, you can say a capping at the ends of polypeptide chain. The N terminus is acetylated and the C terminus is amidated. Okay, same here also they have mentioned. The peptide you can see they have mentioned along with the cappings. So whenever this is written, you can ignore that fact, not required. Just see what is there in between that. Okay, so you should concentrate only A1, A2, A3, A4. Now, second point says that phi psi values, all residues are negative. So it can be concluded that the conformation of the peptide is helix. That means a helix is there. Then a turn is there. That means a turn is there. And then a strand is there. A beta strand is there. So that means they have given three secondary structures. Okay. Now, if you know something about Ramachandran plot, so these are your values for your right-handed helix, beta pleated sheet, all that. Okay. Now for alpha helix, you see both phi psi values are negative. But if you take the beta pleated sheets, for them phi value is negative, but psi value is positive. Isn't it? Similarly, for turns also, you have some negative, positive. So even those should reflect in the Ramachandran plot. So this is your Ramachandran plot, okay? See, over here in this quadrant, beta sheets are allowed. In this quadrant, alpha helix is allowed. In this quadrant, left-handed alpha helix is allowed. So that means you have your alpha helix over here if you have negative phi and negative psi. Now, what has the second statement said? Most of you got wrong in this small thing which they have mentioned. That it is like that for all the residues, all the residues. But if it has some strand, that means beta strand or beta turn, where are their Ramachandran plot confirmations? Tell me, their Ramachandran plot confirmations are not there, isn't it? So that is why, yes, it is acetylation is on N terminus, Janvi. Acetylation is on N terminus and amidation is at C terminus. That is only what happens. Okay. So that is why this statement is wrong. It can be concluded that no, confirmation is only reflecting about alpha helices. There is no confirmation reflection about turn and strand. That is why it is wrong. Fine. The allowed values of phi psi are not valid for short peptides. Why not? It is valid for any dipeptide also which has just one peptide plane. Even for that it is applicable. Okay. And a peptide which has 4 amino acids, it will tend to take a beta turn. The dihedral angles of A2 and A3, they will determine the type of beta turn. So if you look over here in this figure, this is type 1 and this is type 2. You can clearly see that the proline which is present over here, you know that it can take cis and trans, because of which you can see the rotation of the second and the third value, it is different over here. And because of the phi psi values of the second and the third, you get two different types of beta turn. Okay, that's why we can say that the fourth statement was a correct statement. Hence, third option is right. Okay, next question on your screen. Thermodynamics. 3 electron acceptors, X, Y, yeah. Vishal, the second point it just mentions that the peptide has negative phi and negative psi for all the residues. But on the other hand, they are concluding that all the three type of secondary structures are present. But the phi psi value is reflecting only for one type of residue. That's why it is wrong. Okay? Yes, that's right, Mahima. Correct. So the next question, thermodynamics. 3 electron acceptors X, Y, and Z have redox potential of this, and this. So they have given a reaction where the standard cell potential they have given as plus 0.05 volt. Now, which of these electron acceptors are appropriate? If you want, you can use any of these formulas. So, which is the correct answer out of this? X and Y, only X, Y and Z, and only Z. You have to say which of these are electron acceptors out of X, Y, and Z, which is most appropriate. So Sumaya, this may be for subscribers only option also. So better to watch it live in case you don't have the option of watching it later. So you have to use this equation. Delta G0 is equal to minus NFE0, where G0 is the free energy change, of course. N is the number of electrons which is transferred in a reaction. F is Faraday's constant and E0 is standard cell potential. Yes, Pradumesh, your answer is right. Very good. Kantimati correct. Good. So I am getting few options. Come on, you can do it. So are questions getting harder little by little? Yes. Most of you were able to solve the first question. Yes. Mahima, Rimjim, Shaunli, Sunanda, Pratibha, Deepa, Vishal, Bhagya, Ankita. Very good, all of you. Your answers are right. Okay. So see, Simple logic, students, try to understand one thing. This is your equation, right? So, whenever we have a oxidation reduction reaction, right? For example, X is, let's say it was in the form of XH2. When it converts into just X, it is losing to H plus and it is losing 2 electrons. And then YH2, same thing. If Y has to accept that 2 electrons plus 2 protons, okay, then they will become YH2. So that is how you have a reaction which is oxidation, another which is reduction. So a simple logic you look over here, the equation is minus NFE0. That means if your standard cell potential is positive, right? That means your delta G0 would be, so minus and plus, delta G0 will become negative. Now, delta G0 becoming negative, is that favorable or non-favorable? They are asking which out of this will be appropriate. Means they are looking for something which is favorable, isn't it? So tell me, delta G0, if it comes negative sign, then it is favorable or unfavorable? So it is very much spontaneous. Yes, very good. It is favorable, isn't it? That means we are looking for delta G values which will come negative. We are looking for delta G values which will come negative. Okay, now let's keep X over here. How much is X? The cell potential. We have given the redox potential plus 0.15 volt, isn't it? So plus and plus. You have to take the net E0 because one will be for oxidation, one will be for reduction. So totally you have to take this also and the other electron acceptor reaction also. Together you have to take. So if plus and plus for X, what will be the value? Plus only, isn't it? Then for Y, what will be the value of cell potential? Plus and plus, that will come plus only, right? And for Z, what will be the value? You see here minus and plus. So what will be the value over here? You are getting minus, correct? Now delta G will be opposite of that. So if you put plus over here, then you're getting delta G negative, right? Same here also, you will get delta G negative. But if cell potential in Z is negative and you put negative sign over here, then negative and negative will become positive. That is when your delta G will come positive, isn't it? So if your delta G is positive, that means it is not favorable. That electron acceptor is not appropriate. It won't be able to take it. Clear? Everyone clear with this question? Why it is answer is X and Y? Okay, great. So there are many other formulas of delta G, free energy. I am not using all of them today. All right. So this is your explanation, what I just said. I have calculated and shown, but idea is that much. You don't even have to calculate so much. So this is what is coming for Z. Not required. You can solve a big question in a shortcut form also. Okay. All right. Okay. Thank you, Ujjal. Now, next question. In the accompanying figure, reaction kinetics of 3 protein is presented. Protein concentration used to obtain this data, A, B, C. If the catalytic efficiency is defined as kcat by km, okay, if the catalytic efficiency, it is also called as specificity constant. If it is kcat by km, they have given the formula also. Which of the following statement is correct? Yes, KBC, right? Your answers are on the screen. Options are on the screen. Choose the correct answer. So if you are able to solve, means you will get success in CSIR exam. Yes. कौन बनेगा CSIR net achiever? Exactly, Antara. For the previous question, you can just use some logic. Okay, thank you so much, Sampurna, Mahima. Yes, very good, Aditya. Your answer is right. Very quick and prompt. I hope you have solved it the proper way it has to be. Yeah, Bhava Priya, correct. Because solving a question, see these questions, some of them you might have seen before from previous year question papers, but the way to solve it, it should be clear. Because these type of questions, they have a certain protocol. If you follow that, there is no chance of making a mistake. Yes, Devanshi, Manjula, Averi, Rimjim Soni, Kantimati, all of you are right. Pratibha, Harsh, correct. Others, don't worry if your answer is going wrong. I will explain to you the solution. Yes, Nanashri also correct. Very good. Okay, so this is a graphical based question from enzyme kinetics. Yes, Yasira also right. Good. So, graphical based question, for that you have to first see the activity. First of all, what they have asked, kcat by km, right? Now, first let's talk about km. How to calculate km? If you don't know, please note down. Km is the substrate concentration which you require to achieve half of Vmax. Km is the substrate concentration which you require to achieve half of Vmax. Okay. So, what are the Vmax from over here? As you can see, for B, the Vmax, I'll take it as 30 over here. So for B, I'll explain it right here. So for B, it is 30, as you can see. Then for A, it is 20. And for C, if you draw a straight line, it will come to 14, right? So Km is half of maximum activity. So to find out Km, we need to extrapolate from the point of half maximum activity. Now for B, if you take B maximum value is 30, take half of it. Half of it is 15. Where is 15? 15 is here. Touch the B line and bring it down. How much substrate you require to reach half of 30? Half of 30 is 15. That much Vmax, that much velocity to reach that. How much substrate you need? 1. Let's see what is for A. A has maximum velocity of 20. So to reach half of that, that is 10, touch the A line and bring it down. That is also 1. And even C, maximum velocity is 14. 14 by 2 is 7. So that's why 7, from there also, if you touch the C line and bring it down, it will be 1. For that means that all of them, they have the same Km. Km is same for all of them, for all the proteins. Now this order based questions, you have to now depend on kcat. The catalytic activity will depend only on kcat. Now what is the formula for kcat? Vmax is equal to kcat into Et. If you don't know the formula, write down. The total velocity is equal to the turnover number into the total enzyme concentration. Is your total enzyme concentrations given? Very much they are given, right? Yes, very good, Sadhya. After getting the concept, you came to the right answer. Yes, that's right. So your total protein concentrations are given. For A, it is 1. For B, it is 4. For C, it is 2. A chance of silly mistake. The order over here in the graph is BAC. So you tend to use the values 1, 4, 2 in order. But that's not the case. For B, you have to take concentration 4. For A, you have to take concentration 1. Like that. Okay? A chance of mistake if you are not calm enough. So how to calculate the kcat value over here? You have to bring this Et in the denominator. That is kcat is equal to Vmax by Et. So let's do it for B. Now what was the Vmax? What is the Vmax for B? It is 30. What is total enzyme concentration for B? Just now I told you it is 4. What is the kcat for B? It is 7.5. For A, the Vmax value is 20. The enzyme concentration is 1. So the kcat is 20. And for C, the total Vmax value is 14. The concentration is 2, enzyme concentration. So the kcat value is 7. So out of these three, which kcat is highest? A is highest. Yes. After that, which one? After that, B. After that, which one? After that, C. Got it? Everyone clear with this question who were not able to answer? ये आपने बहुत सही बात कहा है काजल राठौर. ग्राफ बेस एंड एप्लीकेशन बेस बहुत हार्ड लगते हैं एंड दैट इज व्हाई यू नीड द करेक्ट गाइडेंस. दैट इज व्हाई यू नीड अ सॉलिड प्रिपरेशन. Superficial classes, unauthenticated, not genuine classes will not give you the correct preparation. Okay, so you need to know what will give you the correct preparation, and that's why Biotecnika is here launching the Lakshya batch. Yes, it's a fresh batch for CSIR net. It starts from 4th Feb. Okay, just uh, uh, one more day to go. So for more information, you can visit us, call us on toll free So what exactly you get over here? You get coaching by the net experts who have a track record of coaching CSIR net and gate exam students appearing for these exams, isn't it? Then you get the portal. Yes, this is a very popular thing at Biotecnika. The portal is a one-stop place for everything. You get all the question papers, ebooks, previous year question papers, ebooks, everything in one place. The recorded lectures, the PPTs which are used in class. So many of you are asking whether these PPTs are available or not. Then we conduct tests, quests, those question papers, their explanation videos. Apart from that, you get academic chat support live from the teacher. You can ask the doubts, right? So everything you're getting in one stop. And there are some perks also if you are enrolling with us now, and that you can talk to our team and ask them what will be best for you. All we want is to make maximum students clear CSI net this year. As you know, there are 3 chances, so do not miss out on this opportunity and make the most of it. It starts tomorrow. The earliest you begin the preparation, you will not miss out on anything, and definitely your preparation will be foolproof. Okay, next question. Ready? All of you ready? Yeah, tell me, Bhumi, what doubt you have. Okay, welcome, Ankita. Okay, your next question is on again on thermodynamics, but a little bit of theoretical. Protein stability is represented as folded into unfolded and back again. So this is K equilibrium. The equilibrium constant. It's a reaction, folded to unfolded, unfolded to folded. Now, thermodynamic parameters, they were determined prior to development of sensitive calorimeters. And this is what is the equation that they have given you, right? Where delta H enthalpy and delta S entropy are standard changes in enthalpy and entropy respectively. So which of the following statement is correct for estimating delta G, delta H and delta S? All these three, which is the correct one for estimating? Determining ratio of folded and unfolded protein at 37 degree, plotting KEQ as a function of delta H, plotting KEQ against delta S, or plotting KEQ against temperature. Tell me, what will be the correct answer? So this question is from June 2017. Yes, very good. Deepa, your answer is right. Very good. Yes, Kantimati, Antara, Nancy, Raumi, Amruta, Nandini, Ankita, your answers are correct. Yes, Ritika Bhagya also correct. So, let me tell you something about the Van 't Hoff's equation, right? So, basically, if we define conditions where heat is transferred, where the heat is transferred is a measure of internal energy, then we have to know that equilibrium is affected by temperature. Okay, and delta H is the enthalpy change for the reaction, where delta H is proportional to how the equilibrium constant for the reaction varies with temperature. So one thing you should know about this endothermic reaction, that the slope is proportional to the delta H for the reaction. The slope what you are seeing, what you have plotted in y-axis is RT, ln, KEQ. In x-axis you have plotted against temperature, 1 by T, right? So in this above case, if it is endothermic, then the slope is proportional to delta H, and in this case, it should be positive. This results in the following completed description of the relationship. That is the equation which I showed you. And experimental data for the relationship between KU and temperature is usually plotted RTLN KEQ versus 1 by T, and delta H is the value of the negative slope. So can you see negative slope is delta H 0? From here you can calculate the value of enthalpy. Okay. Now, let's say the protein chymotrypsinogen has a native to denature, that is folded and unfolded equilibrium that is affected by temperature. Okay. Now, delta H is approximately plus 500 because it gives you positive charge. That means the denatured state is 500 kilojoule mole higher in energy than the folded or native form. Got it? So why is that? If the native form has more and better non-covalent interactions, then energy is required to achieve a state where these are disrupted. So that's why to break a bond you need energy, and that's why it is higher in energy, the denatured form, than the folded or native form. If the native form has sequestered hydrophobic groups from solvent, then exposing them requires an input of energy. Okay, so just remember, a positive delta H value indicates, this is your also a summary statement which may help you in other questions also. A positive delta H value indicates the folded state of the protein is a lower internal energy state than the unfolded state. Okay, so internal energy difference, which is the delta H for protein denaturation, it will vary somewhat with temperature, but is usually positive, which favors the folded state. So enthalpy is not only the contribution to energetics of protein folding, and proteins do unfold at extremes of temperature. Means it is not the only contribution. Okay, so one more thing, if you know delta H, automatically you will know the delta G and delta S because they have a relationship. Okay, I hope that was clear to all of you. Question number 7. So out of all the statements below, from amino acids, from stabilizing interactions, from proteins, from histone complex, so which after the following combinations is correct? Yes, give me the correct answer. This question is from December 2017. Ujjal, you cannot really guarantee on a subunit. What are the chances? You have asked how often questions come from thermodynamics. You can't really guarantee that, okay? You will have to check on the like probability. If it has not, but then also I cannot guarantee. Like if last year it has not come, this year it will come. No subunit you can have guarantee like that. But if you ask me, maybe one in one question paper, one or two in one question paper, or if we take an average, if it will be just one per question paper. In some sometimes you may not even get, but generally one question you will get. Okay. No, Romy, the previous question was just based upon some known statistics, the Van Tovs equations. Yes, very good. Yasira, Antara, Janvi, Ritika, Mahima, your answers are right. Very good. Vishal, Aditi, Aarti, Shaunli also. You have just mentioned C, but I'm guessing you meant 3. Yes, Kratika, Nancy, Averi, Devanshi, right. So let's understand L-threonine and L-allothreonine. They interact identically with plane polarized light. First of all, what are these? These are optical isomers, but they are not enantiomers. Means these are not mirror images of each other. That is why it is L-threonine and L-allothreonine. Had it been L-threonine and D-threonine, then they would have been enantiomer. But then also they would have interacted differently, not identically, because one would bend plane of polarized light to left and another to the right. But here they are saying they are non-mirror images. So how can two non-mirror image structures interact identically with the light? The bending obviously will not be same. That is why A is wrong. Okay, Van der Waals interactions are always attractive. Not at all. There are molecules, if you have some same end of molecules facing each other, they will end up repelling themselves. If you have the opposite ends of molecules coming together, they will end up attracting themselves. So repulsion is also a type of Van der Waals force. That is why we call them Van der Waals interactions. We don't call them Van der Waals bonds or something, right? See, polyproline 2 helix is not stabilized by intermolecular hydrogen bonds. If you see polyproline helix, this is how it looks like. Because Proline, other amino acids, they will have the peptide bond as CONH. You will have C double bond O and you have NH. This is the bond that other amino acids will have between them. But when a proline is involved, the bond before that, that is towards N terminal, the bond will be only CON. There is no hydrogen, as you might be knowing, proline side chain is connected with the own amino group. So there is no hydrogen. And to form secondary structures, to form hydrogen bonds, you need a hydrogen, isn't it? So if hydrogen is not there, from where a proline polypeptide chain, polyproline means a polypeptide chain in which all are proline. From where will you get hydrogen bond stabilizing the structure? Not possible. So that is why it is correct statement. It is said it is not stabilized. That's right. The folding of a protein is associated with an overall positive change in free energy and negative change in entropy. A polypeptide chain has highest entropy. Entropy means degree of randomness. It is very random structure, it is not ordered. But when the protein is folded, order is brought to this protein, and now the entropy decreases. So from high entropy, the entropy is brought down, that is lost, which is correct. It's associated with negative change in entropy. But when a protein folds, bonds are formed between side chain. When bonds are formed, energy is lost. Okay, when a protein folds, energy is lost. Yes, so that is why a polypeptide chain, it has high free energy, whereas a folded polypeptide chain, it has low free energy. That means even free energy is getting lost. So if it is lost, means this also should be negative, but it is written positive. That is why this statement is wrong. D is wrong. Okay. Yeah, that's right, Mahima. Van der Waals interactions are repulsive at a distance lesser than 5 angstrom between atoms. And third one, this is also correct. We know acetylation on histone is associated with loosening of the histone complex from DNA, because acetylation is done on the lysine groups, which were supposed to be having positive charge groups, and that was giving the histone a positive charge, which was able to bind the negative charge phosphate backbone containing DNA. So if the positive charge is made negative with acetylation, then they cannot bind the DNA anymore. And that will end up the histones losing from the DNA complex. That is why here correct option is C and E. Okay. So this is what is your explanation. Next question. Yes, with the protein structure, regarding the protein structure, following statements were made. Hydrogen bonding patterns are given for different helices. Beta turn, how many atoms between the hydrogen bond donor and acceptor? Gamma turn, how many atoms? Parallel sheets have evenly spaced hydrogen bonds at an angle. Which out of this is correct combination? A and C, A and B, C and D, B and D. So we know that to stabilize secondary structures, we need hydrogen bonding. So which hydrogen bonding are there in alpha helix or 310? Yes, Kratika, Pradumesh, Antara, Nancy, Vishal, Ankita, Ritika, Alia, Shubham, Nandini, Rimjim, Nanashri, all of you are right. Very good. Yes, Deepa, Ujjal, Mahima, Kantimati, Megha, Priyanka, Devanshi. So if you look very closely, this is actually supposed to be a very easy question. Because in alpha helix, you don't have n to n plus 3. No, you have n to n plus 4. Isn't it? CO of n to NH of n plus 4. That's right. So A cannot be right. If A cannot be right, none of this is the option. So you have to select from 3 and 4. C and D and B and D. Definitely D is correct. Parallel sheets have evenly spaced hydrogen bonds which bridge the strands at an angle. So if you check over here, This is parallel beta sheet, anti-parallel. Anti-parallel has straight bonds, whereas parallel has angled hydrogen bonds. That is why fourth statement is right. Now let's see why the third statement and the second statement, which is correct or wrong. Let's analyze. See, this is the peptide plane. If we consider this as the first, second, third, fourth, and fifth, sixth, right? So, how many hydrogen bonds are present between the bonding with 310? This is what is a beta turn. Beta turn has 4 amino acids, that is 1, 2, 3, 4 in the turn, where the fourth amino acid. So first I will take it as N, fourth I will take it as N plus 3. First is joining with the fourth one, that is NH group of the fourth one, which is N plus 3. That means this is how a beta turn forms. Now, how many atoms are present over here? If we start from the hydrogen bond donor, means acceptor, and to the donor, how many atoms are going in the backbone? We are not counting the side ones. We will only count the ones which are in the backbone. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. That's correct. Beta turn indeed has 10 atoms in the backbone present in between acceptor and donor. Now gamma turn, it is formed by N to N plus 2. So N to N plus 2 means you will have this as the structure, right? Because this is first amino acid, second and third. The bonding is from CO of the first amino acid to the NH of the third amino acid. So what is the bonding? How many atoms are there? 1, 2, 3, 4, 5, 6, 7. But what is written in the previous one? It is written there are 6 atoms between hydrogen bond donor and acceptor, which means that this is not correct. B is from December 2019. Following observations are made on a 30 residue polypeptide. Unordered structure is observed in water. These are observations. Unordered structure is observed in water. These are observations, these are not options, okay? So based on these observations, you have to tell which out of these statements correctly attributed to above observations. So which will be the correct answer? Are you able to hear me properly? Are you all able to hear me properly? Okay. So I hope you did not miss out on anything. Everything was clear. Yes, Yasira, very good. Your answer is correct. Antara, Pradumesh also, answer is right. Read it carefully. You have to read all the observations, okay? Yes, Kantimati, Rimjim also right. Very good. Yeah, Devanshi also correct. Okay, Mahima. Yeah. So, yes. Manjula, Bhagya, Nanashri, Nandini, Alia, Sunanda, Nancy, Averi, Ankita. Very good, all of you. Indeed, it is first option. Peptide is entirely composed of D amino acids and is amphipathic. First thing you should know about the unordered structure, they have said it lacks a fixed 3D structure. The like charges attraction or unlike charges repulsion. So it is there because of which it is not there because of which it contains it does not have any fixed 3D structure, and it contains high proportion of polar and charged amino acid residues. Now they said it takes a helical conformation in a low dielectric constant. What is dielectric constant? Very short, it is a measure of polarity. If you have high dielectric constant, means in polar environment, like in water. If it is low dielectric constant, it is in non-polar environment, in presence of hydrophobic groups. So amino acid sequence affects alpha helix stability. Any polypeptide chain cannot take up an alpha helix. There are certain amino acid criteria which will lead to the formation of an alpha helix. So the statement was unordered structure is observed in water. That's fine because in water it's a area of high dielectric constant. Okay, but when it comes to low dielectric constant, a helical structure is formed. So how come there are two different structures in two different dielectric constant? Means in low dielectric constant where there is non-polar groups, the non-polar groups are trying to expose outside. As you can see here, the side chains are exposed outside in alpha helix, isn't it? So that means it is a correct statement. It is amphipathic, means it is amphipathic. We can conclude that the protein is it's amphipathic, consisting of both polar and non-polar groups. Okay. Second option says the peptide is resistant to degradation by proteases. That definitely means it is a D amino acid, because we know that in our body, in most amino acids, they are consisting of L amino acids, and proteases and all, they are very much specific to L amino acids. So if it is resistant to degradation, it definitely means it is D amino acid. Red blood cells are lysed by the peptide. I can't say it is giving much of information for the conclusion over here, because anything can cause the lysis of the membrane. Beta mercaptoethanol has no effect on peptide structure. Beta mercaptoethanol, it is a reducing agent. So it breaks disulfide bonds. When it is saying it does not have any effect, it means that there are not much disulfide bonds. It means that cysteine residues are not present that much. So definitely this statement cannot be correct. The peptide is rich in disulfide bonds. Peptide is entirely amino acids. L amino acids, again no, because how will it explain the degradation by proteases? So definitely even this is not correct. That is why the correct answer is 1. Very good. Many of you got it correct. Okay, the last question of the session. Pay attention, all of you have to answer it correctly. Okay, I want participation from all of you. In the form of stress response, bacteria synthesize a group of proteins called stress proteins, such as DNA K, DNA J, GROL, GROS, GRPE. Now, DNA K is an ATP binding protein that will attach to the newly synthesized polypeptide in conjunction with DNA J. So, which out of the following statement correctly states a step in the subsequent process of protein folding? So, the affinity of DNA K increases upon hydrolysis, or DNA J is a exchange factor, or ATP hydrolysis is required for phosphorylation, or ATP hydrolysis is required for the phosphorylation of GRPE. Come on, get it correct. So this is an information based question. You need to know the concept, then only you will be able to answer this question. If anyone attended the decision making webinar of easy, tempting and trap, so I would suggest this question is totally informative. If you have not studied protein folding, then do not even try to attempt such question. It can be a trap question for you. Till now, I haven't got the correct answer from anyone. Yes, now your answer is right, Ritika. Very good. Yes, Nida Parveen, Devanshi, very good. Okay, no problem. Very good decision, Sunanda. Sunanda Singh says skip. That's a very good decision. I really liked it. So it's the first time anyone has said like that. Okay, I can't solve or I'll skip this question. So you have learned the art of decision making, which is really important. And if anyone has not seen the previous webinar, please see it and learn the art of skipping. You have to take a decision. I'm going to attend this or I'm not going to attend this. Okay. Yes, Sharon, Rajat, Explore Life Science, Vishal, all of your answers is correct. Now let us see the explanation. See, the process, first of all, it starts with DNA J binding to an unfolded protein. This is the protein folding process with some chaperones, help of some chaperones. Okay. So DNA J binds to the unfolded polypeptide chain, and that will recruit DNA K. DNA K has ATP bonded with it. Now, once the DNA J and DNA K with the ATP has bonded, the ATP gets hydrolyzed. So you can see here, along with DNA K, you have ADP. So when it is having ADP along with the DNA K, it has more affinity to the unfolded protein. Affinity increases with hydrolysis, okay, of DNA K. Then comes GRPE. It is a nucleotide exchange factor. Nucleotide exchange factor will take out the DNA, take out the ADP from here, and it will input an ATP. It is an exchange factor. So no, DNA J is not exchange factor. DNA J is recruiting DNA K. Here exchange factor is GRPE. It stimulates the release of ADP. Okay. So once again ATP is bonded with DNA K, it loses the affinity with the protein, and that's how the protein is released. In this entire process, there is a chance that they will fold up. If they fold up, well and good, native conformation. If they don't fold up, then they go into another system of chaperonin, which is the GROL and GROS. So let's analyze the options over here. The affinity of DNA K to the polypeptide increases upon hydrolysis of ATP to ADP. Yes, this is the correct answer. Because when the DNA K hydrolyzes ATP to ADP, upon binding with ADP, it is having a conformational change, which is making it having more affinity towards the peptide. So first is the right option. Okay. Second, DNA J is an exchange factor. No, it is not. It helps to recruit DNA K. ATP hydrolysis is required for the phosphorylation of DNA J. Not at all. It is required with DNA K. It is required for phosphorylation of GRPE. Not at all. It is not associated. Clear? Everyone clear with this question? Why option 1 is correct? So tell me honestly, how much did this session benefit you? Yes, did you learn concepts? Some of you got some questions right, some of you got some questions wrong. But was it helpful for you or not? Do you feel a bit confident that okay, you are ready to at least go out in the exam? And the people for whom it seems all new, don't worry, we are here for you. If you are coming in with us in the Lakshya batch, then we take the guarantee that we will help you clear CSIR with flying colors. Okay. Do we have to register separately for tomorrow's session? You can just talk to the team for that. Nitya. Okay, that's really good to know, Devanshi. I'm very happy for that. All right. I skipped one wrong, rest correct. That's amazing, Sunanda. So that means you are very well prepared. UT thermodynamics very nicely. Thank you so much, Ujjal. I'm glad that you felt so. Okay, so stay tuned for tomorrow's session, everyone. It's going to be another interesting session. What are the units you teach on the platform? Ujjal, I teach unit 1 and unit 5 and a few other topics in the classes, in the regular classes. So tomorrow, our unit 2 specialist, expert CSIR net unit 2 faculty member is going to teach all. You are going to learn a lot of concepts from unit 2. Okay. So with that, I will definitely take your leave. Thank you everyone for joining in. So please contact our team if you're interested in the Lakshya batch. With that, thank you everyone. Good night. Join in for another session tomorrow. Bye.